Hello and welcome. My name is Katherine Gallagher, and I am a professor of surgery and microbiology and immunology at the University of Michigan Medical School. Today, I'll be taking you through a small tour of some of the research that my laboratory has worked on, mainly in the inflammation and tissue regeneration in vascular disease space. There are multiple cells, both immune and structural, that play a role in vascular inflammation. And as many of us know in the cardiovascular, cardiometabolic realm, inflammation is one of the main pathologic processes that is important in pathology and disease. And a lot of the diseases that we study are due to excess vascular inflammation. While there's multiple cell types that contribute to this excess inflammation that we see in vascular disease, macrophages are one of the primary immune cells that our group and others have studied because they play a major role in vascular inflammation and in mediating vascular inflammation in many different disease states. Inflammation, though, is not all bad. You need normal inflammation to occur following any kind of insult or injury, and it's a necessary process in order to heal. However, when you have excess inflammation, is really when a lot of the pathology that we look at begins. So, normally, as it relates to macrophages, you have pro inflammatory macrophages that come into sites of injury or sites of damage, and they perform vital roles where they phagocytose debris, produce necessary cytokines that are important for recruiting other cells to the area of injury. And then these macrophages transition to an anti inflammatory phenotype where they help clean up the wound or the tissue or the area of injury, secrete other growth factors, which bring in additional cells that are important for angiogenesis and vasculogenesis and remodeling of the tissue. And this is actually a necessary and important process in any kind of vascular injury. However, in a lot of the pathologies that we look at, in aneurysms, atherosclerosis, non-healing wounds, diabetes, there is an excess of these pro-inflammatory macrophages that do not transition to this anti-inflammatory phenotype. So they remain sort of locked, if you would, in this pro-inflammatory state and are unable to transition. So our lab really looks at, why are these macrophages locked in this pro-inflammatory state? Why can they not transition to this anti-inflammatory state? So this initially led our group really to look at what is regulating this macrophage inflammation and this sort of cell plasticity that we see, where these macrophages should be able to transition between a pro-inflammatory phenotype and an anti-inflammatory phenotype. So this led our group 15 years ago, when our lab got started, to look at epigenetic alterations, which are basically changes in gene expression patterns that can control cell plasticity and cell phenotypes. So this was highly relevant to us because we were looking at, why are these macrophages consistently making these inflammatory genes and not making these anti-inflammatory genes. I have this slide up just to recognize that there's multiple different types of epigenetic alterations that can occur. There's histone modification, there's DNA methylation, there's RNA interference like microRNAs, and there's additional ones that are not even mentioned on this slide. So all of these are different processes that occur in cells that can essentially regulate the expression of inflammatory genes. Our group mainly studies the one on the left, histone modifications, and how alterations in histone biology can control gene expression and inflammation in disease. So, just to kind of take a deeper dive now into these histone modifications that our group looks at and how that might regulate inflammation. It's important to remember that DNA is kind of tightly wound into these nucleosomes. You have these histone tails that are present as part of this complex. And based on these are all different amino acid residues shown here. And based on the specific mark or the specific area where you have activity occurring, you can either close down the chromatin or open the chromatin and allow transcription factors to bind and either turn genes on or off. And it's very site specific. So that's kind of what I really want to point out in this slide. Is that based on where so in this case, we're talking about methylation. So on the left of the slide, these are all enzymes that add methyl groups to this K4 residue. And so when you add a methyl there, you open the chromatin, you allow transcription factors like NF-kappa B and other common transcription factors to come in and bind. You turn gene expression on. Whereas at this other site, K27, these are all enzymes that essentially can remove methyl groups or add methyl groups in various cases. But if you add methyl groups to this site, to this K27 site, you essentially close down chromatin, transcription factors can't bind, and you turn off gene expression effectively. So this is just an oversimplified definition or an oversimplified schematic to show you conceptually that kind of based on the very specific position that you have these enzymes acting. And again, I'm just showing you methylation, but there's multiple different types of enzymes that can act in these areas and to basically affect gene transcription and particularly inflammatory gene transcription. So why is this and where is this clinically relevant? And I just put up a couple of examples that we have studied, but obviously it's been studied in lots of different contexts and in lots of different arenas. But some of the ones that we'll talk about briefly today are in tissue regeneration and tissue repair, particularly in wound repair and especially in diabetic patients, in the context of sepsis, in the context of COVID-19 in patients affected with cardiovascular disease, as well as in abdominal aortic aneurysms. I'm not talking about atherosclerosis today, although it's been well studied in that arena as well. So one of the histone modifications that our group studied very early on, and I'm just going to use it as an example to show you how it plays a role both in tissue regeneration and in other processes, is known as SETDB2. And SETDB2 is a histone methyltransferase, if you remember from that initial schematic. It adds a methyl group at this particular K9 residue and effectively turns genes off. So its role is to add a methyl and turn genes off. Very early on, and I'm not showing you a few slides that kind of show we observationally found that inflammation was increased in diabetic wound macrophages, both from human tissues that we isolated as well as from mouse models of diabetes. And we did some various arrays that identified that SETDB2 may be relevant in human macrophages associated with non-healing wounds in diabetes. So this led us to look at this molecularly and to kind of look at what the mechanisms for this might be using some of our murine models as well as some human tissue. But one of the first things we did is we created a mouse, a SETDB2 floxed mouse, that we crossed to a lyse2 Cree, or effectively made it so that SETDB2 was not present in macrophages and only in macrophages or myeloid cells in this mouse model. And then we looked at classic inflammatory cytokine gene expression that you would see in diabetic wounds, where you see elevated TNF, IL-1B, IL-6, IL-18, all of these very inflammatory cytokines, IL-17. And in this example, we isolated macrophages from the wounds of these mice that were deficient in SETDB2 in macrophages and found that inflammatory gene expression was significantly increased when you were missing this epigenetic enzyme called SETDB2. And these are just various different ways of showing it, looking at both the gene expression themselves, looking at protein expression, and then also looking when we perform a chromatin immunoprecipitation assay, looking at the promoters of these various genes. In this example, I'm showing you IL-1B, but looking for that K9 methylation, since we again know that this enzyme increases K9 methylation. When we look at human tissue as well as mouse tissue, we sure enough found that the SETDB2 was decreased in the diabetic setting. So really important, and you can see that it increases in a normal setting. And so in normal wound macrophages, you have this spike in SETDB2 around day 5, which is around the time when you want to decrease inflammation in diabetic or in normal tissue rather. So you want SETDB2 to go up at the right time, turn off inflammation in these macrophages, and restore normal homeostasis and normal wound healing. What we found in diabetic models, both human and mouse models, is when we look at the macrophages from those settings in diabetes, they do not have adequate SETDB2 at any time point, and especially not at those critical time points around day 5 or so when you want to turn these phases off. So this led us back to how is this being induced in wounds? So we know that SETDB2 is low in the diabetic setting, and we know that we need it to be increased. How do we increase it? What is controlling it upstream? So through a series of experiments, which we're not going to detail here because we're just trying to kind of showcase some of the highlights of this, we identified that type 1 interferons via JAK-STAT signaling seem to be controlling the SETDB2 enzyme. And what we had in the setting of diabetes was not enough interferon, beta, type 1 interferon, to upregulate the SETDB2 in the diabetic wound macrophages. Whereas in normal tissue, you have a spike in interferon, you have appropriate JAK-STAT signaling, and then shutting down via SETDB2 of inflammation in these diabetic wound macrophages. So this was very important for us and very relevant. And we were looking at how can we increase or regulate levels of interferon beta. Also, we were looking at, was there something wrong with JAK-STAT signaling in this setting? But this essentially, when we were looking as well at what downstream does SETDB2 control, we identified that one of the common pathways that we see increased in diabetic wounds and in diabetic patients is uric acid. It's a metabolite that is significantly increased. It's strongly associated with inflammation, but is unclear why uric acid is increased in diabetic wounds. Well, through again a series of experiments that we won't go through here, we identified that SETDB2 was controlling gene expression of an enzyme that regulates the xanthine to uric acid conversion in macrophages. And so when SETDB2 is present, it essentially can shut down this pathway. When it's decreased, as we see in the diabetic setting, it will result in increase in uric acid. And so this was important because it shows the link that is also well described in lots of other diseases and lots of other processes between metabolism and epigenetics. And those two things are really strongly associated, where metabolism can often regulate epigenetic pathways, and epigenetic pathways can often in reverse regulate metabolic pathways. So this was, again, I don't want to belabor or go into a lot of this data here. This is sort of data overload. And if you have any interest in these specific stories or looking at all of the data, many of the papers are referenced below for where you can find more specific information on these stories. But essentially, again, through using these mice that we created and through using diabetic tissue and human tissue, we were able to see how this gene that controls this enzyme that regulates the xanthine to uric acid conversion was also controlled by this SETDB2. So now I'd like to switch gears and show you another histone modification that our group has looked at that does something slightly different, where we have modification at that K4 site, where it's a different enzyme that we're going to talk about called MLL1, can add methyl groups here at this K4 site and essentially allow transcription factors to bind and turn on inflammatory gene expression. So kind of an enzyme that has sort of an opposite role, if you would, of SETDB2, which is serving to serve as a brake and decrease inflammation at the appropriate time. This enzyme revs up inflammation and increases it. I'm not going to show you a lot about this story, but just wanted to mention that this enzyme plays a role in promoting inflammation and is again necessary for normal inflammation and normal wound repair, but in the setting of diabetes becomes pathologic where exists in excess, and you have excess MLL1 present at time points long after inflammation should have resolved, where you cannot clamp down or decrease the amount of these inflammatory cytokines like IL-1B and TNF alpha. And so you wind up getting excess. And this is just showing that you don't have enough of this MLL1 very early on when you would need it to promote inflammation or early inflammation, which is really important for getting rid of pathogens, recruiting other cells that are necessary for the wound. But you have excess present in the diabetic setting in late wound macrophages. So again, just kind of highlighting that you have this dysregulated epigenetic program, and so we're looking at ways that you can kind of get rid of MLL1 at late time points in diabetic wounds. So much of this work and a lot of other work that I haven't shown you led to a scientific statement published in Circulation, which is shown here, which essentially, in addition to going over clinical management of diabetic wounds and non-healing diabetic wounds, also laid out some of these scientific foundations that I've talked about here in looking at sort of newer, novel, maybe more cell-specific directed ways to target these non-healing wounds in cardiovascular patients and in diabetic patients in specifically, where we're looking at ways of modulating some of these epigenetic programs in cells, and namely in macrophages, to see if we can reverse some of the chronic inflammation that we see there. This is just highlighting that although I'm kind of showing you a small snippet of the story, this is obviously a very complex story, and there's multiple other factors at play. Diabetic wounds don't heal for many reasons, including macro and microvascular disease and lots of other cellular dysfunctions shown here. Again, we're really kind of focusing on a very specific area that our lab focuses on, but please keep in mind that there's inflammatory cell dysfunction across the board. There's multiple other vascular cells that are dysfunctional, as well as many other factors sort of upstream and macrovascular disease that's also affected in these patients. The pathogenesis, again, is very complicated. There are multiple other structural cells and other immune cells playing strong roles here, including fibroblasts, keratinocytes, and other cells discussed in this larger review. So some of the next steps as it relates to tissue regeneration in our group have been, and we've been focused on this for the last 5 years, have been using nanocarriers that target specifically these macrophages and are trying to incorporate inhibitors or drugs, small molecule drugs that can target some of these epigenetic pathways. So in the case of MLL1, we're trying to decrease the MLL present in the macrophages in these diabetic wounds. In the case of SETDB2, we're trying to upregulate some of these upstream pathways in the macrophages, the interferon, JAK-STAT pathways that are upstream in these in the diabetic setting, and try to upregulate the SETDB2. So again, that's why it's important for us to understand these molecular pathways, because it really depends on what's going on as to whether you want to up or down regulate something. So in the setting of sepsis patients, it's been long established, and this is just to kind of showcase another use of some of the work that we had been looking at, and how we discovered it was also relevant in another disease that commonly affects cardiovascular patients. It's well established that in patients, when they've recovered from sepsis, even if it's 5 years after recovery from a major ICU level sepsis episode, these patients have an impaired immune response, and they are unable to respond normally to pathogens or other insults because they can't mount an appropriate immune response. Again, this is even years after recovery, not in the immediate recovery period. And so someone in our group was very much interested in looking at this from an epigenetic angle. And when we took human monocytes that we isolated, or monocyte macrophages that were isolated from peripheral blood, we found that in these patients, again, these were several years out from recovery from a massive septic episode, that MLL1 was indeed decreased. And if you remember, I just showed you in the tissue regeneration space that MLL1 was very important for promoting inflammatory cytokines and was important for inflammation. It was pathologic in the setting of diabetes. But what we found through this study, and again, there was a lot of molecular work done in various mouse models and otherwise, but I'm just showing you a figure from the human tissue that MLL1 was decreased in macrophages from these post sepsis patients. And so it seemed to be important to promote a normal inflammatory response in these patients. So in this patient population, we might be looking at ways to increase MLL1 in the macrophages so that they could mount an appropriate response and respond to bacteria or other pathogens once they had recovered. So again, just to change gears and show you another story and how epigenetic enzymes are relevant, and to go back to SETDB2, which was one of the original ones that I had shown you. COVID came in 2020, 2021, shut down a lot of the labs in the States, including at University of Michigan. We fortunately, because we were an immunology-based lab, we remained and continued to turn our attention toward looking at COVID. Since we're also a translational science lab, many of the people working in our lab were also working in the hospital, seeing many of the COVID patients with diabetes and other cardiovascular disease were not doing as well when they developed severe COVID as patients who did not have diabetes or other cardiovascular disease. And a lot of this was attributed to these large cytokine storms. Again, some of these same inflammatory cytokines that I've been talking about throughout this presentation, TNF alpha, IL-1B, IL-6, seem to be at dramatically high increases in these patients that had diabetes and COVID and were dying in the ICU. So again, one of the people working in our lab, James Melvin, took a very, very strong look at this to see sort of why this was happening and to look at it again from an epigenetic angle, because we also knew that macrophages and monocyte macrophages, right, were one of the primary drivers of inflammation in general. And so we sought to identify whether there were any epigenetic alterations that we saw in macrophages from human patients with COVID-19 that we were able to recover. And sure enough, the strongest change that we saw was with SETDB2. So if you look on the left here, we took CD14 cells, which are monocytes essentially isolated from the blood. And these are monocytes that have a strong preponderance towards becoming macrophages if they go into tissue. And we took and isolated these monocytes from healthy donors and from COVID negative and COVID positive ICU patients, and found that in patients that were strongly COVID positive in the ICU, their monocyte macrophages had decreased levels of SETDB2. We then also used a murine model of coronavirus, MHV-A59, in our mouse models. And then we looked at both in vitro bone marrow macrophages as well as in vivo isolated macrophages from these mice, and found that SETDB2 levels, sure enough, were lower in the coronavirus infected animals. Through a series of other tests, and again, I'm not showing you all the data, and I don't really want to focus on specific data. I just kind of want to make the point that, again, in these settings, SETDB2 seem to regulate inflammatory cytokines that were NF-kappa B responsive inflammatory cytokines in response to infection with coronavirus. So when you got this MHV virus and we isolated your macrophages, you had an excess, you had lower SETDB2 and an excess of inflammatory cytokines. And then sure enough, when we looked at diabetic animals, it was even greater and even more dramatic response, probably because you were starting with a lower level of SETDB2 to begin with. And so you had less inflammatory cytokine. When we looked at blood from these patients, because again, remember from the tissue regeneration story, we had some concept that interferon beta via JAK-STAT signaling may be driving the SETDB2. When we looked back at this, we found that interferon beta was also at decreased levels in plasma from the patients in the ICU that were COVID positive that had diabetes. And then sure enough, when we took human monocyte-derived macrophages from non-diabetic and diabetic patients, we found lower levels of interferon beta in these. In our mouse model, when we took both normal and diabetic macrophages that were infected with this coronavirus and gave them interferon, we could upregulate SETDB2 nicely in the diabetic patients to a larger, or in the diabetic mice rather, to a larger degree than in the normal diet. And then again, not to get weighed down in data, but this also, in using our mouse model, appeared to be regulated by an interferon JAK1 STAT3 model. So it seemed to be STAT3 in the setting of coronavirus, and we could modulate various cytokines, including IL-1B, TNF, and IL-6, based on levels of interferon and looking at this downstream pathway. Now, importantly, as it relates to the diabetic patients, again, I told you they had less SETDB2 to begin with. Our question was, can we increase the SETDB2 in this coronavirus infected in macrophages in the mice by giving them interferon treatment? And sure enough, we were able to do that. When we gave them interferon, we were able to upregulate SETDB2, and we were able to downregulate the cytokines to try to decrease the cytokine storm in these macrophages. So I think this was kind of a nice use of some of the data that we had already generated and looking at how some of these epigenetic regulators, and in this case, SETDB2, seem to control macrophage-mediated inflammation. You can see here for the coronavirus story, it seemed that SETDB2 was already downregulated in the setting of diabetes. When you had coronavirus, it was kind of a double hit, and it decreased SETDB2 even further, making this, dysregulated inflammatory response that much greater in this setting. So again, looking at maybe specifically targeting that pathway in the setting of coronavirus. So I'm going to end and discuss kind of one last enzyme and how it plays a role, and this is going to be in aortic aneurysms. A lot of this work was done by Frank Davis in our lab, who continues to study the role of epigenetic enzymes in aneurysms. But through, again, a series of arrays, we identified that JMJD3, which is another epigenetic enzyme, this one is a histone demethylase, so it removes a methyl group, but by doing that, it essentially will turn gene expression on or allow transcription factors to bind. But we identified in human aneurysm tissue that this enzyme seemed to be strongly upregulated. So we investigated what it was doing molecularly. Again, when you have an increase in JMJD3, because it's a demethylase, it's removing methyl groups, and then it opens those promoters and allows transcription factors to bind, turning gene expression on. So one of our first forays here was using, again, human tissue. We knew that some of these common NF-kappa B mediated inflammatory genes, IL-1B, TNF alpha, and others, were all increased in a human abdominal aortic aneurysm tissue. That's just showing you this here. Since we had other arrays that had pointed to JMJD3 being increased, we looked in our human tissue samples and found, sure enough, that JMJD3 was increased in human aortic aneurysm tissue as compared to aortic control tissue. And you can see over here that JMJD3 was indeed increased, particularly in the myeloid cell component, the myeloid macrophage cell component in human aneurysm tissue. We also did single cell sequencing on human aortic aneurysm tissue as well as control tissue. And this is just showing you the different cell populations that we see. When we looked at monocyte macrophages, or sort of the myeloid cell population, we could see that JMJD3 expression was significantly increased in this cell population. And you can see that nicely shown over there in various subsets of monocytes as well as macrophages. There's a significant increase in JAM-JD3 in the human tissue, in the human monocyte macrophages in aneurysm tissue compared to controls. And sure enough, when we do a pathway analysis and look at these macrophages that are positive for JAM-JD3 and have strong levels of JAM-JD3 expression compared to the cells that do not, the inflammatory response, as well as response to IL-1 and cytokine-mediating signal pathways, are some of the strongest upregulated pathways in these cells. Again, suggesting the very strong link between JAM-JD3 and inflammatory response. We use multiple models of aneurysms in the mice, and this is just showing you in one of these models, the ANG2 model. When we look at JAMJ83 in cells isolated from these aneurysms in these mice, you can see that, and in macrophages specifically, that JAMJ83 at the gene expression level was indeed increased increased in these Ang2 mice. That corresponded with increases in all of these inflammatory cytokines, IL-1B, 23, and NOS. And then when we performed CHIP assay, again, remembering that this was working at the K27 methylation mark and was a demethylase, right, removing methyl groups from there, you can see that at the promoters of all of these inflammatory genes, you can see a decrease in K27 methylation in the aneurysm group. Now, importantly, right, because we're a translational lab, we're looking at, okay, we know there's a lot of increased JAMJ83 in the macrophages in aneurysms. How do we get rid of this? Because we want to get rid of the JAMJ83, see if by controlling inflammation, we can perhaps improve aneurysms and decrease them. And so we used pharmacologic drug JSK-J4, which essentially can decrease JAMJ83. We found that indeed, as you can see here in the Ang2 mouse model, it prevented aneurysm formation. And when we isolated macrophages from these cells, it decreased inflammatory cytokines. I'm just showing you here IL-1B and TNF alpha. And then when we did a genetic model, which is again, we created a JAMJ83 floxed mouse, cross it to the lice M3. So again, where you just should have JAMJ83 deficient in myeloid cells, we saw a reduction in aneurysm formation. And I'm not showing you the pictures of that, but they can be seen in the paper reference below. So you do decrease aneurysm formation, and sure enough, you also decrease the cytokines, those inflammatory cytokines in macrophages isolated from this model. So less IL-1B, less TNF alpha. So again, thank you for listening. I just, in summary, would like to say, that these were just kind of some highlights of some work that we had done in looking at ways that modulating these epigenetic enzymes in the setting of macrophages could control inflammation, and particularly in diseases where chronic inflammation is high, like aortic aneurysms, like diabetic non-healing wounds, and other processes that we discussed today. And thank you to all of the lab members that had contributed to this work, both past and present. Thank you.